After a pretty long hiatus, we are finally back, and this time we'll be talking about physiology. So, we have already talked about some topics from anatomy, we have talked about some topics from biochemistry, and obviously, we will be delving more into those subjects later as well. But today, we talk about a topic from physiology, right? So, as far as the human physiology is concerned, we target in this very specific video the endocrine system of human body. And obviously, since that's a huge unit, a huge topic, so obviously we will be breaking that down, and we won't be obviously dealing with the entire thing today. But what we will be dealing with today is the basic introductory stuff, and with that, along with that, a little more advanced stuff related to the mechanism of hormone action. Right? Now, a disclaimer here. The disclaimer here is that the mechanism of hormone action about hormones, this is much more elaborately discussed, or much more elaborately, the scope is much more in case of biochemistry. So in both biochemistry and physiology, we talk about endocrinology, but their scopes and perspectives are different. As far as the chapter that we are about to discuss in this video is concerned, mechanism of hormone action, that is much more elaborate in biochemistry. But we talk about physiology, so we'll talk about only the basic stuff related to the mechanism of hormone action. But obviously, when we talk about the same in biochemistry, we will be talking in depth or a little bit more. We'll add a little bit more. Obviously, it's the same thing. It obviously does not change, but in biochemistry, we'll be like discovering a little more about the mechanism of hormone action. But like I said, we'll first be dealing with some basic stuff, then we'll be going on to those things, like advanced things. So let's get on with the very basic things at first, and that is hormones. So what are hormones? We all know this, so let me get this out straight very fast. So hormones are basically, chemical messengers, and obviously they are secreted from very specialized cells, which we say to be glands, right? And these chemical messengers, what is their job? Well, their job is extremely varied, but one thing that we can all agree is that their job is to stimulate specific target organs or cells and stimulate the action of those organs or cells. So that's in one way defining hormones. Now, what are the different types of glands which secrete these hormones? That is the first thing that we talk about in very briefly. So let's talk about that very briefly. The glands, what are their type? Very simple. First is obviously the endocrine glands. What are endocrine glands? We all know this very well. Still, it's worth repeating. Endocrine glands are nothing but the glands which actually release their secretions, namely hormones, into the bloodstream directly. They are known as the endocrine glands. Right? Second type of glands which we might say is the paracrine glands. So what are the paracrine glands? Paracrine glands are just like the endocrine glands, but unlike the endocrine glands, these paracrine glands do not release their hormone directly into the blood, but rather they release their hormones into the extracellular fluid, or ECF. Extracellular fluid. So they will release their secretions in the extracellular fluid, and from there these hormones, they will essentially migrate to the target glands, or not target gland, target, yeah, it might be a gland as well, we'll see to that later, but target organs. It will migrate there and it will stimulate the action of that very particular organ. So one thing that we can all agree is that paracrine glands are secreting local hormones. Because if you depend on extracellular fluid to get your job done and not something as universal as blood, then obviously your target gland or target organ or target cells are close to the source of the hormone. So an example of paracrine gland will be, or a paracrine hormone rather, will be essentially something like somatostatin. Somatostatin in the, what can we say, pancreas. So somatostatin in pancreas is a very good example of paracrine gland, right? So extracellular fluid, this is where the secretions go and essentially what happens. Then obviously, see, we are scheming through these, okay, because these are very basic stuff and we are not that interested in these things. These are very basic things. Then obviously we have something called the autocrine glands, and autocrine glands, automatic, auto itself. So autocrine glands are glands which will secrete hormones or which will secrete something which will act on themselves. So they are essentially, the source and the target are the same. Okay. An example of autocrine hormone can be something like platelet activating factor, all right, or PAF, platelet activating factor, released from, it's actually released from endothelial cells, that is the blood vessels, the wall of the blood vessels which are made by the cells, they are known as the endothelial cells. So vascular endothelium, they actually release platelet activating factor, and vascular and platelet activating factor, platelets also in the blood, so figures. So that's how autocrine glands work, all right. And then obviously we have actual neurons which are releasing hormones, and they are known as the neuroendocrine gland, or neuroendocrine cells to be more specific. And obviously they are what? They are neural tissue, neural cells, neurons, which are releasing hormones from their axon terminal. So the hormones which are secreted by these neuroendocrine cells, they are produced in their cell body, go through the axon, released through the axon terminal. And what's an example of this? Very famous example, all the posterior pituitary hormones. Because as we know, posterior pituitary is not an actual synthesizer of its hormones, rather a storage for the hormones secreted by hypothalamus. So something like antidiuretic hormone, or vasopressin, which is also known as vasopressin, or oxytocin, right? So these are examples of hormones which are neuroendocrine in nature. Okay? Okay. So that's a very basic overlay. That's just something to start the chapter with, just the introductory part. Now again, just like we have an a mechanism of dividing the glands into these, we also have a classification for hormones. So hormones can be classified in obviously various ways. We can classify hormones, but one of the most famous methods of classifying hormones is, let me first write it. So one of the most famous, it's not famous, but it's very widespread, and that is according to the chemical nature of the hormones. So hormones can be divided into various groups according to their chemical nature. So there are some hormones which are steroids. There are some hormones which are peptides. And these are the two major groups of hormones. But there are others as well, other categories which are not steroid, neither peptides. They are said to be thyroxine derivatives. This is another classification. Did I say thyroxine? I'm sorry. Tyrosine, I'm sorry. Tyrosine, the amino acid, their derivative. So something like, for example, steroid, it can be something like cortisol, maybe, right? Or it can also be something like aldosterone, right? So these are examples of steroid hormones, right? We'll deal with these individual hormones later in later discussions when we talk about in depth the endocrinology of different glands. Then we have peptides, and obviously we all know insulin, it can be growth hormone, right? It can be adrenocorticotropic hormone, etc. Okay, just to name a few. And these which do not fall either steroid or peptide category, these tyrosine derivatives, they are the thyroid hormones like thyroxine, right? Or this is T4, it can be triiodothyronine as well, T3, right? Or calcitonin as well, right? Thus, having talked about the classification of hormones on the basis of the chemical nature, let's now quickly talk about the classification of hormones on the basis of the location of their receptors. Yes, that's also a very, very important classification. So, essentially, see, hormones can also be classified on the basis of, as I have said, the location of their receptors. So what are the classifications then? The receptors of the hormones can be located like in the normal scenario, they can be located on the plasma membrane itself, or they can not be located on the plasma membrane and they can be intracellular, that is within the cell. So they can be on the cell membrane or they can be within the cell. So essentially the hormones which have their receptors on the plasma membrane, they are basically hormones which are not soluble in lipid, because cell membrane, we all know that its chemical nature is lipid in nature, right? So if the hormone is not soluble in the lipid, then it obviously cannot penetrate the cell membrane. So it has its receptors on the plasma membrane so that the hormone does not need to get inside the cell. Whereas the hormones which are lipid soluble have their receptors within the cell. Okay, so they are lipid soluble. So obviously steroid hormones, they will have their receptors within the cell, intracellular, whereas the peptide hormones will have their receptors on the plasma membrane. Also, let me tell you another thing. These hormones, the tyrosine derivatives, they also have their receptors intracellular. Okay, so they are also lipid soluble. But there is another key difference between the tyrosine derivative hormones and steroid hormones as far as their location of receptors is concerned. Both of these have intracellular receptors, but these intracellular receptors can again be two types, right? The intracellular, within the cell, it might be in the cytoplasm or it might be in the nucleus. Okay, so both of these locations are intracellular, right? In cytoplasm, here is a twist. Steroid hormones, they have their receptors in the cytoplasm, whereas the tyrosine derivatives, these have their receptors in the nucleus. So both of these classification of hormones have intracellular receptors, whereas the peptide hormones mostly have receptors on the plasma membrane. Okay, so this is another classification of hormones, right? And there can be other classification as well. So hormones can be general hormones which act on target organs or target cells which are a little far away from their site of formation, like most normal hormones. But they can also be local hormones. So local hormones are generally, as the name suggests, the source and the target are very close to each other. For example, as I have said, somatostatin, the paracrine secretions, they are local hormones, right? Whereas most of the other hormones are generally acting hormones, right? So that is another classification. And as I have already mentioned, hormones can also be lipid soluble or they can be water soluble. So that is something. So see, the classification of hormones are all very overlapping. They are all very overlapping. It's not that the classification criteria are extremely different from each other. They are very much the same, overlapping, right? So the chemical nature, this is the classification. On the basis of the location of the receptors, this is the classification. On the basis of the site of their action, local or the general, that is their classification. Or on the basis of their solubility, they can be lipid soluble or water soluble, right? So that is all about classification of hormones. So that essentially almost completes our introductory part of hormones. Now, just a few more things we need to know before we can move on to the juicy area. So, what are those few more things? We need to have a little bit of an idea about what essentially an agonist is and an antagonist is. So bear with me now. See, an agonist. What is an agonist? An agonist is nothing but, it is obviously a chemical substance which is very much similar structurally to a hormone, right? And can actually activate the hormone's receptor. Okay? Oh, I didn't see the camera. I'm sorry. This is the thing. So, an agonist, what is that? Essentially, an agonist is a chemical substance very structurally similar to a hormone, right? And so structurally similar that that agonist can bind to the receptor which is meant for the hormone. So that is an agonist. On the other hand, an antagonist, as the name suggests, antagonist, it is what? It is also a chemical substance which will bind with the hormone receptor and will in turn prevent the physical binding of the hormone whose receptor that is. So that is an antagonist. So an antagonist is what? It is essentially almost the same as an agonist. See, here is the thing. An antagonist is almost the same as an agonist. Both will bind with the receptors meant for their hormones, right? But the thing is, an agonist when bound to the hormone's receptor will produce the effects. It will produce the effects of the hormone, but an antagonist will not produce the effects of the hormone. In fact, it will actually negate the action of the hormone, because the actual hormone is not able to bind with its receptor. The antagonist is bound with it, so the action of that hormone is hindered. But in case of an agonist, the action is not hindered, and that is very much, self-evident from their names. So that is the concept of an agonist and an antagonist. Okay. Another important concept that we need to know before we delve into the actual mechanism of hormone action is upregulation and downregulation of hormones. Now, it's pretty simple. It's pretty simple concept. You see, upregulation, that is nothing but the concentration of the hormone on decreasing increases the number of receptors meant for the hormone. That concept is called upregulation. So if the concentration of the hormone decreases, the number of receptors for the hormone, I'm sorry, I don't know what's happening to me today. I'm very messy with the camera today. So please pardon me. Hormones concentration on decreasing stimulates an increase in the receptor concentration, or the number of receptors meant for the hormone. This is called upregulation. Whereas downregulation is just the opposite of that. That if there is increased hormone concentration, there will be a decreased number of receptors. Okay. So the reason for this, I personally, I'm being very honest with you, I could not understand the reason for this. So I really need to do a little bit more research about this. But essentially my textbook refers to this, or rather reasons this out by saying that essentially when the concentration of the hormone increases, the number of receptors initially increase, but later they are for some reason endocytosed, and that reduces the number of receptors. But I could not understand why they are endocytosed in the first place. So pardon me, but I really need to do a little bit more research about this. So I probably will release something about this if I find out in my website. Yes, that's true. I have actually opened a website, and that is essentially for all the additional stuff that I miss out during these discussions. I'll be posting it there. So that's also my god, this is a terrible way of announcing a website. Just in the middle of a discussion I'm announcing it, but the link will be down in the description below, and if I find anything, it will all be there. But do visit my website, okay? Just for seeing all that I could have done. Anyways, it's pretty weird now. So that's about upregulation and downregulation. Okay, so we talked about the basic introductory stuff. We talked about hormones. We talked about the different classifications of glands, hormones, the concept of agonist, antagonist, upregulation, downregulation. And that wraps up the introductory part. Now let's get on with the main thing, which is essentially what we are meant to do here, and that is the mechanism of hormone action. Okay. So while talking about the mechanism of hormone action, the first thing that we need to learn about is a special type of a protein. Why do we need to learn about a special protein before knowing about the mechanism of hormone action? Let me tell you. See, the mechanism of hormone action, it is to know that, to understand the different mechanisms by which hormones act, we need to learn about second messengers and G proteins. because in most mechanisms, G protein is actually associated with that mechanism. So what is this G protein, how it acts? Let's quickly give that a run through. So what is G protein? So obviously, as the name suggests, is some form of protein, right? What protein? G protein is what we say to be hetero trimeric protein. Okay, it is a hetero trimeric protein. What does that mean? Hetero means different, not homo, hetero. Trimeric, 3 subunits. So it has 3 different subunits as a protein. Now let me give you a disclaimer here. G protein can be of 2 types. One is the heterotrimeric kind, and another one is known as the small G protein. Small G protein. So it can be a heterotrimeric G protein, or it can be small G protein. As far as the mechanism of hormone action is concerned, we are only concerned with heterotrimeric G proteins, not small G proteins. Small G protein are linked to other things, not mechanisms. So we won't be talking about them. Okay? Okay, so from now onwards, G protein, when we'll be talking about that, we'll be meaning heterotrimeric G protein. Okay? Okay. So what is this heterotrimeric G protein? It has 3 subunits. Okay? So suppose this over here is the cell membrane. Okay, so this obviously is the cell membrane. Okay, I'm showing the bilayer. Okay, okay. So as we know, this is probably this is a known concept that every hormone has a specific receptor, right? So G proteins and all these other mechanisms mostly that we are going to talk about, they are all linked with all those hormones which are not soluble in lipid. Okay, because the hormones which are soluble in lipid, they can easily pass through the cell membrane, bind with the receptors, and then bring about changes in the nuclear DNA and lead to transcription of their specific genes, leading to formation of different proteins. So we'll talk about that later in a little bit detail, but that is essentially what they do. But the bulk of this chapter lies on the different mechanisms which are employed by the hormones which are not soluble in lipid. Okay, that is where G protein comes into play. Okay, so G protein is actually nothing but a heterotrimeric protein which is having the function of translating the external signal into some form of an intracellular effect. That is what G protein is. So suppose there is a receptor. Okay, this is essentially I don't know if you know about this, but most peptide hormones have a 7-pass receptor. Okay, so 1, 2, 3, 4, 5, 6, 7. So their polypeptide tail actually passes through the receptor or through the cell membrane 7 times. That's why it's known as 7-pass receptor. Receptors are also proteins, by the way. So this is essentially a receptor. Okay, okay, and this is the hormone binding site of the receptor, right? Now this receptor, just like it has this extracellular part, it also has an intracellular part over here. So this intracellular part of some receptors are bound to or linked to G protein. Okay, a G protein. What is a G protein? As I've already said, it is heterotrimeric. So it has 3 subunits. What are the subunits? One subunit is known as the alpha subunit. One subunit is known as the alpha subunit. Okay. Another subunit is known as the gamma subunit. So suppose I place it over here, gamma, and another subunit is known as the beta subunit. So alpha, beta, gamma, I don't know why I built up so much tension here. It was just alpha, beta and gamma. But the diagram here is a little bit more important than it seems. See, alpha and gamma, these are the two subunits which are linked to or which are attached to the cell membrane here by covalent bonds. Alpha and gamma are the ones which are anchored. Beta is attached to gamma. So this is something that you should know about. Okay? Okay. Another thing that you should know about is that this alpha subunit is linked to what we say to be a GDP molecule. It is linked to this GDP molecule. Okay? Okay. So this is essentially how an inactivated form of G protein looks like. No problem up to now? Okay. Now, suppose a hormone or a ligand which is meant to bind over here. So the hormone for this receptor comes and binds with this receptor. What this will actually do is the receptor will, so obviously the hormone binds to the receptor. So now the receptor will actually activate this G protein. How that happens? Let's talk about that. Binding of the hormone now activates the 1, 2, 3, 4, 5, 6, 7. So now that will actually activate the alpha subunit. And what it will do is, it will release the GDP and will bind with a GTP. So a GTP will in turn bind with the alpha subunit. GTP. Okay, I'm sorry it's getting a little messy maybe, but this is essentially what happens. Alright. So GDP is given off, GTP binds. This is essentially a signature of the G protein being activated. Okay, this is essentially G protein getting activated. Now the beta gamma subunit or the beta gamma complex, that remains. Okay, it remains. Now what will happen is this alpha activated alpha subunit, it will essentially now move on to the, it will detach itself from the cell membrane, and it will move on to other intracellular or maybe other proteins or other intracellular hormones, or maybe the hormone is also present in the cell membrane. It might be any of these things. So essentially it will move on and it will promote the action of that intracellular hormone, in turn which will lead to activation of other hormones, which in turn will do some other things, and this will go on, and thus we say that a cascade will be formed. A cascade of intracellular signaling. Okay, so a cascade is actually formed here, right? Now, so see, we are only, this is not the actual mechanism. We are not talking about the mechanism of hormone action here. Bear in mind. So don't actually have this impression that he's not talking about anything that's basic thing or anything like that. We are not talking about the mechanism right now. We are only talking about G protein and how it works. All right. The mechanism, the actual mechanism, we haven't even started yet. So that's it. The cascade of intracellular signaling will essentially start. Okay. Now, what happens to the beta gamma complex? That is not a simple answer. Essentially, it will depend on what kind of a cell or what kind of a gland or what kind of a hormone you're talking about. You see, in some cases, it is the beta gamma subunit or the beta gamma, not subunit, the beta gamma complex, which will participate in the cascade of intracellular signaling. Sometimes it is the activated alpha subunit which will take part in this cascade of intracellular signaling. Sometimes they will cooperate, right? Sometimes they will have, cooperative action on some form of cascade action. See, the actual function of G protein is to start the cascade. Cascade means a series of steps occurring one after the other, right? So G protein's work is to sense this external signal and to transduce that or translate that into an intracellular stimulation, hence starting this cascade of intracellular signals. What essentially are the steps? What essentially is this cascade? That is where the bulk of mechanism of action of hormones lies. And this will depend on what hormone are you talking about, right? For some hormones, this cascade might be the activation of cyclic Gmp, essentially going on to activate protein kinase A and then phosphorylation of different inactivated proteins, activating them and then leading on to their function. For some, it might be the release of calcium, right? For some, it might be the activation of phosphatidyl, or rather the splitting up of phosphatidyl inositol bisphosphate into diacylglycerol and inositol triphosphate. So we'll see all to that later. They are the bulk of mechanism of hormone action. That is where we get specific. But here what we are trying to demonstrate is what is G protein and how it acts. Okay, this is not the actual mechanism by which hormones act. This is just a single step of the mechanism. Okay. So sometimes it might be this. Sometimes it can be the gamma beta or the beta gamma complex which can do its work. All right. So that's how different things occur. So now let me actually give you an example where it is the beta gamma complex which mediates the action. So for example, if I talk about, suppose, the acetylcholine, ACH, it's written like this. Vagus nerve secretes acetylcholine. Okay, this acetylcholine, when it acts on the muscarinic receptors in heart, when they act on this, what will happen is the beta gamma the beta gamma complex will get activated. Okay, so when the vagus nerve will actually secrete acetylcholine, which will act on the muscarinic receptors in heart, the beta gamma complex will get activated, which in turn, what it will do, the beta gamma complex will cause the opening up of potassium channels in the cell membrane, which will in turn lead to K plus efflux K plus efflux from the heart cells into the surrounding medium, extracellular fluid. So what will that cause? That will cause hyperpolarization, leading to what we call to be IPSP, that is inhibitory post-synaptic potential formation. So inhibitory post synaptic potential formation, which will in turn decrease the heart rate. Okay, now bear with me. I'll try to explain this. So essentially, see what happens. Vagus nerve. We all know about the vagus nerve, the 10th cranial nerve. We all know about it. It releases acetylcholine. Acetylcholine is not an actual hormone. It's more of a neurotransmitter, right? Acetylcholine will bind with the muscarinic receptors in heart. So the receptors which bind with acetylcholine, they are known as the muscarinic receptors. So it will the walls of the heart, they actually have these muscarinic receptors. So acetylcholine will bind with these receptors in heart. Okay, pretty simple up to this, no problem. Simple facts. What acetylcholine will do is, from the vagus nerve, the vagus nerve is essentially, it has this function of decreasing the heart rate. It has this function. How? This is the procedure. So muscarinic receptors in heart, so it will be something like acetylcholine binding with the muscarinic receptor, leading to activation of not the alpha but the beta gamma complex. So this is an example of the beta gamma complex being activated. So the beta gamma complex on being activated, see what I said, it will lead on to the cascade of intracellular signaling. So here that cascade, I don't know if it's a cascade of actions leading to potassium channels opening up, or if it is the beta gamma complex itself directly leading to the opening up of potassium channels. But something we can agree upon is that this essentially somehow directly or indirectly leads to opening up of potassium channels in the heart or in the cardiac cells. What will happen if the potassium channels open up? As you know, potassium already has a tendency of exiting from the cell, right? It is a sea of sodium outside, and cells are essentially bags of potassium floating in sea of sodium. That is how Dr. Najib describes it. I really love that way. So potassium is more inside, less outside. So obviously potassium exits. So there is a potassium efflux due to opening up of potassium channels. What this will cause is hyperpolarization. If sodium had entered the cell, or if potassium exits the cell, so if there is a K plus efflux, what would that lead to? Hyperpolarization. Isn't that true? If, suppose, what does the sodium potassium channel itself does? Think about this. What does the sodium potassium pump does? ATPase pump do? It essentially pumps sodium out, keeping the potassium in. So it takes in potassium, throws out sodium. That leads to depolarization. So if there is potassium which is thrown out, that will lead to hyperpolarization. My explanation was really bad, so don't go for it. In turn, what you might do is read the action potential chapter yourself from the neurophysiology part. All right, we'll be dealing with that later. So this essentially leads to hyperpolarization. Hyperpolarization, which means that, what is this? An inhibitory post-synaptic potential is formed. So a post-synaptic potential can be excitatory or inhibitory. These are all linked to neurophysiology. We'll deal with this later. So in this case, due to hyperpolarization, it is the inhibitory post-synaptic potential which is being formed, inhibitory. So it will decrease the heart rate. On the other hand, when the vagus nerve secretes acetylcholine, which will bind with not the muscarinic receptors in heart, but the muscarinic receptors in the smooth muscles of stomach. So if it is not the heart but the stomach which is in case, with which the acetylcholine binds, then it is the alpha subunit which is activated. So if this is the muscarinic receptors, I'm abbreviating this, of the stomach that we are talking about, that will lead to what? Alpha subunit, alpha activation of the G protein, which in turn will do exactly the opposite of what was done here. That is, it will cause the closure of K plus channels. It will cause closure of the K plus channels, leading to, this was hyperpolarization, this will be depolarization. And if it is depolarization, we all know that you are essentially moving in the correct direction. Depolarization, we all know that is the first step in the formation of an excitatory postsynaptic potential. So in place of an IPSP being formed, it will be an EPSP, excitatory postsynaptic potential being formed. Excitatory. So what will it excite the muscle to do? What the muscle is supposed to do, that is contraction. So contraction of stomach muscle, that is the smooth muscle of the stomach, okay, or the gastric smooth muscles. So this is how, see, how amazing that the subunit which is being activated of a G protein is deciding what the action at the ultimate case will be. That is amazing. That is really amazing. So in case of the heart, it was the beta gamma complex being activated, which in turn led to the opening up of potassium channels, leading to efflux of the potassium, leading to an inhibitory postsynaptic potential formation, leading to decrease in heart rate. And that is why we say vagus or acetylcholine actually decreases the heart rate. But in case of stomach, acetylcholine, what it does, or vagus, what it does, increases contraction of the gastric smooth muscles, because it is the alpha subunit of the G protein which is activated. Okay. So that is that about G protein. So what happens after that? What happens after the G protein is activated, and how does the G protein essentially start the cascade of that intracellular signaling pathway? What happens? So for knowing that, we need to know about, just like the G protein, we learned a little bit about that, we need to learn a little bit about second messengers. So let's learn a little bit about second messengers. So what are these second messengers? These second messengers are, you see, the activation of these G proteins that leads to activation or the formation of second messengers. Remember what I said when I was defining hormones? Hormones are chemical messengers. They are the first messenger. So hormones, they are bringing a message, but at the cell membrane, they are blocked off. They are said that, no, you cannot enter. So what can the hormone do? What the hormone does is, it calls to its friend G protein, who happens to be inside the cell or at the interior surface of the cell membrane. The hormone calls G protein and says, my friend, it's now your job to carry forward the message which I was bringing. G protein says, yeah, sure, okay, I'll do your job. So what G protein does is, it will call on to another set of chemical substances which are known as the second messengers. And G protein says to the second messengers that now it is your job to move on to the deeper parts of the cell, the more interior sections of the cell, find those proteins which can do the job of my friend hormone, and find them, phosphorylate them, and make them do that job. So that essentially is where second messengers come into play. So second messengers are another part of the cascade which moves on or which goes on as far as the intracellular signaling is concerned. So the concept of second messengers, or why second messengers is required, is because hormones, they could not enter, or the kind of hormones which require second messengers, they cannot enter the cell membrane, through the cell membrane. So they call on to G proteins, which in turn lead to the formation of these special chemical substances which are known as second messengers, which just like the first messenger will move on inside the cell and will deliver the message, will lead to the formation of those specific proteins which can perform the function which the first messenger, the hormone, intended but could not unfortunately do so because of its barrier from the cell membrane. So that essentially is the concept behind second messengers. Okay. So some examples of second messengers. Let's first give some examples of the second messengers, right? So what can be examples of second messengers? An amazing example is Cyclic Gmp. Another example can be, just like AMP, Cyclic Gmp. Then we have examples like calcium. We have examples like IP3, inositol triphosphate, or DAG, diacylglycerol, etc. So these are all second messengers, and these are mostly, almost all of them, almost, not all of them, not all of them, but almost all of them are activated by some G protein. So G protein, after getting the signal from the receptor, after itself being activated, it will lead to the formation of these second messengers. Not all of them. For example, cyclic GMP is not linked to G protein. We'll see to that later. But mostly what happens is they get the message from G protein. G protein leads to or stimulates the synthesis of these intermediate chemical substances, the second messengers, which in turn will lead to the formation of some other proteins or substances, etc., chemicals, which will in turn carry on the function which was intended at the first place, right? So see, this is step by step. That's why this is a cascade. That's why we call this a cascade. That's why this is a cascade of intracellular signaling. Intracellular signaling is not one step or two steps. It's a cascade of steps, right? This will activate this will activate that will lead to the formation of that, so on and so forth. All right? So that is the concept of second messengers. But we need to know a little bit more about second messengers. In fact, we need to learn a little bit about the features of second messengers. It's raining outside. It's a beautiful weather outside, but we are stuck here. Anyways, so pardon the noise from the rain, by the way. What are the features of these second messengers? So let's very briefly talk about the features. The first feature is that, see, this is a very basic concept which I'm going to talk about here. Whenever in physiology or biochemistry there is a cascade of steps, whenever there is something leading to something, something leading to something, this, that, and an entire signaling pathway or an entire pathway is formed, most of the times in subsequent steps there will be amplification of that signal, or there will be an amplification of that effect. And this is a very basic concept. This is something that is seen in many cases, not just intracellular signaling. Intracellular signaling is what we are describing here, what we are talking about here. But we will see this in many other enzyme actions, many other physiological processes, that whenever there is a cascade, many times there is an amplification of the signal or of the physiological effect thanks to the second messengers. Okay. And another feature, although this is not very general, I would say this is general because most of the times this is what happens, is that second messengers in most cases, as we will see later, they will activate some form of kinase. And as we know, kinases are phosphorylating enzymes. So what kinases will do is they will in turn phosphorylate some protein, which will in turn activate the protein, which will in turn function, or which will in turn act on some other thing, and this will go on. So essentially the action of the hormone, the first messenger, that is brought about by the second messengers. How? Mostly by activating some kinase which will phosphorylate the protein, thus activating it, and that protein will then carry on its function. Right? This is what is seen mostly. But then again, we also have the other hormones which are steroids or lipid soluble. They do not need second messengers, right? So what they will do is they will themselves enter, bind with their receptors, form a receptor hormone complex. That complex will in turn move on to the nucleus and bind with some specific portions of the DNA. Some specific portions of the DNA, which are known as hormone responsive elements, right? And that will essentially lead to some form of transcription, leading to formation of specific mRNAs, leading to translation and formation of the target proteins. So see, why I brought that up, although we'll discuss about that later, why I brought that up right now is, here in case of the second messenger system, kinase is activated, inactive proteins to active protein, done. The proteins will keep acting. But in case of the steroids or the other hormones which are lipid soluble and do not need second messengers, they will first bind with the hormone, they will then bind with the hormone responsive element of the DNA, they will then lead to the transcription of the specific parts of the DNA leading to specific mRNAs, which in turn will be translated to form those specific proteins, which in turn then will form, or rather will function. So these second messengers activated proteins, these are very, very rapid in their action. They are extremely rapid in function, whereas the others, that is the steroid ones, they are pretty slow in comparison. Okay. Now another feature which I think you can all appreciate is that the number of second messengers, when you compare that with the extreme diversity of functions which the hormones perform, when you compare that with the number of second messengers, we can all agree that there are only a very few second messengers. Very, very few second messengers are present. And even then, they will lead to a wide array of functions. How? How is this possible? How is it that many hormones will have the same second messenger but will then again have different functions? How is this? This is very simple. The answer is very simple. The answer is, it is not the second messengers themselves which will act as the proteins and different other chemicals and will in turn function. The function of the second messengers is only to activate the different kinases, leading to activation of different proteins, formation of different proteins, and they will then do their job. So the proteins which are activated are diverse, but the second messengers themselves are a few. So who cares if they are a few? They get the job done. So that is the reason why, even though there are only a very few second messengers, there is a huge array of functions which are carried out by different hormones even when they are using the same second messenger. Okay, so that is the reason for that. Now, having talked about second messengers and having talked about G proteins, let's now talk about the real thing, which is the mechanism of hormone action. Up to now, we were gathering the components which will facilitate our understanding of mechanism of hormone action. Thus, we studied about G proteins. Thus, we studied about second messengers. Now, let's see how they correlate and how the actual mechanism is brought about. So, before going into that, let's first classify the mechanism of hormone action and see how many classifications can be drawn about as far as the mechanism of hormone action is concerned. So, the mechanism of hormone action, it can be through what? It can obviously be through the most, the thing which we'll be discussing the most is, that is through second messengers. But it is not the only mechanism through which hormones act. This is one of the mechanisms through which hormones act. The others being something like through intracellular receptors, which is something which we have also talked about a little bit. That is through intracellular receptors. As I have already mentioned, second messengers are needed by those hormones which are not lipid soluble, right? So they have their receptors on the plasma membrane. But what about the hormones which can pass the cell membrane? They don't need second messengers, because there the first messenger itself can cross the barrier, right? So they act through intracellular receptors, right? There is another mechanism, although it is not one mechanism, it is like groups of mechanisms which are not by second messengers, they are also not by intracellular receptors. They are different others, like they do not fall in either category. So it might be something like autophosphorylation of hormone receptor complex, or it can be something like through tyrosine kinase or mechanism, or it can be something like JAK-STAT pathway. So we'll see to all these later. So for now, we'll only say that there are others as well. Alright, bear with me. I know this is extremely misleading, does not explain anything, but bear with me, we'll talk about this later. Alright? Alright. For now, we are very much interested in the first category, which is through second messenger system, because we learned so much only to go through these hormone systems, that is through second messengers. The others are easy enough, although the other section is not that much, but this is extremely easy. So let's go through them one by one, starting with the second messenger system, that is through the second messengers. Okay, so as you can all see, I've already given the heading over here, the mechanism of hormone action, and I've also already put out our first topic to talk about as far as mechanism of hormone action is concerned, and that is by second messenger. The first second messenger that we'll be dealing with is cyclic AMP. So how, remember I had exemplified under second messengers, I had exemplified cyclic AMP. So how does that happen? Let's talk about that. So for that we need a little bit of a diagram going on, right? So let's draw it. So for example, this over here is the cell membrane. Okay, no problem. All right. So this over here is the cell membrane, and obviously in the cell membrane we have a receptor. Now I'm simplifying the receptor. We are not drawing the 7-pass receptor because it will be a little more complicated. We are simplifying it as something like a wedge shape. Okay, so this is the receptor. So when a hormone binds with this receptor, suppose this is the hormone, fits into the wedge of the receptor, right? What this will do is, this will in turn activate what? Remember? Yes, G protein. This will activate the G protein. So the G protein should also be somewhere over here. So suppose it is over here. So suppose this over here is the G protein. Okay, so this is the G protein. So the hormone receptor, the hormone when bound with the receptor will signal the G protein, and that will in turn lead to the activation of the G protein by hydrolyzing, or rather giving off the GDP and binding with the GTP. Okay. Now when bound with the GTP, the G protein gets activated. What will the G protein do in this case is, it will in turn activate another enzyme located in the cell membrane itself. So this over here is the adenylyl cyclase. So G protein, upon being stimulated or being activated itself, will lead to the activation of this enzyme, which is adenylyl cyclase, in the cell membrane itself. Adenylyl cyclase, why that is activated? That is activated because this is the enzyme which can kind of catalyze the formation of cyclic AMP from ATP. So what will happen now? There is ATP in the cell. By the action of adenylyl cyclase, that will form cyclic AMP. So thus G protein leads to the formation of the second messenger, cyclic AMP, by activating adenylyl cyclase. Okay, done. Now cyclic AMP, we are describing through cyclic AMP, we are describing the mechanism of hormone action through cyclic AMP. So what cyclic AMP will do? Very simple. Cyclic AMP, remember what I had said when we were talking about second messengers? Most second messengers act by what? Activating other protein kinases. So cyclic AMP will actually activate a protein kinase. What is the name of that protein kinase? It is known as cyclic AMP dependent protein kinase, or in short, protein kinase A. Protein kinase A. So what happens over here? So this is the inactive protein kinase A. Very sad. So sad, inactive protein kinase A. But cyclic AMP, what it will do is lead to it getting activated. So now it will be what? Active protein kinase A. So it is now very happy. Alright. So cyclic AMP turned inactive protein kinase A to active protein kinase A. And this is the active protein kinase A, or rather it is the active protein kinase A which will carry on the functions of different of the hormone. So how it will do that is, it will essentially phosphorylate different target intracellular proteins. As I've already said, that what it will do, the active protein kinase A will now lead to some phosphorylation of intracellular proteins, right? And by phosphorylation, those proteins will be activated, and hence what will happen is they will lead to an actual cellular response. So an actual cellular response. So cellular response was the intention of the hormone. The hormone wanted the cell to respond in a particular way. The hormone wanted the cell to perhaps stop proliferating. The hormone perhaps wanted the cell to stop secreting. The hormone perhaps wanted the cell to increase its secretion, something like that. And that is essentially achieved by this. So this is how by different pathways, obviously we actually encapsulated the phosphorylation of intracellular proteins and that different part. We are not elaborating on this, and directly leading to cellular response. Okay, so that essentially is how this occurs. Now let me tell you something. When I was talking about G protein, I said that alpha subunit is activated by GTP being bound to the alpha subunit. But how is it inactivated? Very simple. When the GTP is hydrolyzed back to GTP, then the alpha subunit again returns back to its inactive state, and again then it will go on and bind to the interior of the cell membrane where it is supposed to remain along with the beta gamma complex, and thus the inactive form of G protein returns. So that is essentially how it functions, or that is essentially how it is acting. I had forgotten to mention that, so pardon me for that. But there is another thing. See, G protein, it has another form. Okay, so there is another form. So for example, this is the G protein which is stimulating adenylyl cyclase and thus leading to the cascade being formed. So it is a you might say positively enforcing G protein which we are seeing here. But there is another type of G protein which is the G inhibitory type. So there can be GS, which is the G stimulatory, or there can be GI, which is G inhibitory type. So the inhibitory G protein will inhibit adenylyl cyclase. So just like the GS over here was activating adenylyl cyclase, GI can inhibit adenylyl cyclase and will stop the entire cascade. Alright, so obviously we need the stoppage of the cascade at some point. Hormone, it remains bound to the receptor. Does not mean that the cellular response should keep going on and on and on. Right, at some point it must stop. How it stops? One of the ways is G inhibitory type of protein, of the G protein actually inhibiting adenylyl cyclase and stopping the entire process. But there is another way, or rather there are other two ways by which this can be stopped. Right? What are they? They are, now listen to me very carefully. See, over here we are seeing that ATP to cyclic AMP is being formed, right? There are some forms of, you might say, enzymes which are known as phosphodieesterases. Phosphodiester bond breaking enzymes. So what they will do is they will actually inhibit the formation of cyclic AMP from ATP, hence stopping the entire process. So these are phosphodiesterase enzymes can actually inhibit the formation of cyclic AMP and will stop the entire process. That is one way in which the cell can inhibit or terminate the signal. Another way is these are phosphorylated intracellular proteins, right? So in case of enzymes which can remove the phosphates from the proteins, if there is any such enzyme, then the activated protein will again turn back to its inactive form, right? That is exactly what happens in phosphatases enzyme. You know, phosphatase enzymes, they are the different enzymes which can remove the phosphorus or the phosphate bond. So they will again lead to the termination of, or rather the dephosphorylation of the intracellular proteins, hence terminating the cellular response. Right? Right. So that is how the termination of signal occurs. Now, there is another thing which I need to tell to you. So, hold on. So, another way in which the G protein coupled receptor intracellular signaling pathway is terminated is by arresting the G receptor, or arresting the receptor itself, not the G receptor, arresting the receptor itself. Okay, so see what we talked about. We talked about the G inhibitory protein, which is another variant of the G protein, which can in turn lead to the termination of the signal pathway. We talked about phosphodiesterase enzymes, which can in turn break the phosphodiester bonds and hence lead to the termination of the pathway over here by not letting ATP convert to cyclic AMP. We talked about phosphatases, which can dephosphorylate the intracellular proteins which are already phosphorylated, hence deactivating them, hence terminating the signal. But there is another way. And that other way is something which I should have talked about when we were talking about G protein itself. But anyway, since we are talking about how the pathway is terminated, we can talk about it here as well. So see, even if the hormone is bound, the hormone or the ligand is bound to the receptor for indefinite amount of time, the cellular response cannot go on indefinitely. There has to be a time when that needs to stop. How that is stopped is by these things, and another pathway is, let me talk about it. You see, the hormone receptor, if I draw it over here, so it is something like this. This is the hormone, this over here is the receptor. So, on prolonged binding with the hormone or the ligand, what will happen is specific kinases will actually arrive at the site. Specific kinases will arrive at the site, and what they will do is they will essentially phosphorylate the receptor. They will essentially phosphorylate the receptor, these different kinases. Now, after phosphorylation by the different kinases, what will essentially happen is this will lead to the arresting of the receptor by a special kind of a protein known as Aristin protein. So let me draw it up first. So this is the hormone, that is the receptor, and this obviously essentially are the phosphates, so phosphorylated. So this will essentially lead to another special kind of a protein called Aristin. This is known as Aristin, Tin. So this protein, as its name suggests, Aristin, will arrest the receptor. Now what will happen is, this receptor, what it was doing, it was essentially activating the G protein. Due to the Aristin protein blocking off that, the G protein will not be stimulated any further and the entire pathway will stop. So we saw G stimulatory, G inhibitory protein, that one controlling the pathway. Then we saw phosphodiesterases, which can essentially stop the pathway here. Phosphatases can stop the pathway over here, and Aristin protein can stop the pathway here itself. But obviously this is only done under physiological conditions when prolonged exposure has occurred for the receptor towards the hormone. So we don't want a prolonged one. So we don't want cellular response to go on and on indefinitely. So to terminate that, this actually occurs. Okay. Now that was all about cyclic AMP. Okay, so that is the entire pathway of through cyclic AMP. Now the next one that we talk about is through cyclic GMP. But there is a twist in this. Okay, the cyclic GMP we can fit in over here itself because it's very small. There is not much to know about this. So what is that? Through cyclic GMP. All right. So cyclic GMP, the pathway for cyclic GMP is very much similar to that of cyclic AMP, with only one major difference. And the major difference, as I think I have already said a little bit about, is that cyclic GMP does not incorporate the activity of G protein. So there is no G protein activity. What does that mean? This G protein at individual cyclase, ATP to cyclic AMP, then it was occurring, right? But in case of cyclic GMP, if I can draw it very fast, it will be something like the hormone, the receptor, right? This is directly linked to the guanylyl cyclase. Okay, so if this is the cell membrane, then the guanylyl cyclase, for example, I'm drawing it over here, guanylyl cyclase, this enzyme is directly linked to the receptor, the intracellular part of the receptor, rather than a G protein which will activate guanylyl cyclase. Guanylyl cyclase itself is incorporated in the intracellular part of the receptor, and thus the rest is same. CGMP will be formed, cyclic GMP. Cyclic GMP will then essentially lead to the formation of an inactive protein kinase G to activated protein kinase G. Remember, this is protein kinase A. What was the other name for protein kinase A? Cyclic AMP dependent protein kinase. So that is protein kinase A. So obviously protein kinase A will not be the same protein kinase, or it won't be the protein kinase for cyclic GMP. So what is the protein kinase for cyclic GMP? The protein kinase for cyclic GMP is protein kinase G, or as we say, cyclic GMP dependent protein kinase. And rest is the same, that it will lead to the phosphorylation of different proteins, activate them, and they will bring about the different functions. Okay, so I think this video has gone on for too long right now. So I think we'll end it right here. So we talked about a lot in this video, and we talked about the introductory stuff about hormones, obviously. We talked about the different, G proteins, the stuff about G proteins. We talked about the stuff about second messengers. Then we started the mechanism of hormone action. We talked about the cyclic AMP pathway. We talked about the cyclic GMP pathway. Next discussion will complete the mechanism of hormone action. There we'll talk about the other secondary messenger systems and essentially the intracellular receptor system and the other hormone mechanisms which do not follow either of these two. And then we'll essentially complete the entire chapter. So I hope this was a very long one, and whatever I have missed or whatever I'll be missing out or whatever extra that can be incorporated, I'll be now uploading to my website, and the link will be there down below. It's a very it's a bad website. I know it's not a professional level website, but I've tried to do something, and I've tried to put on some information as much as I can, and more information will be subsequently delivered over there as much as I can and as much as I can research out. So do visit that website in the description below, and I hope you can understand whatever I try to discuss over here, and hope you found this discussion worthwhile. Thank you very much. Thank you for sticking out. This is Medman signing out. Thank you very much. Peace.